module talks about basic immunology, the study of the structure and function of the immune system. It aims to provide a brief overview of some of the main concepts of immunology as they relate to immunization. However, a detailed review of immunology is beyond the scope of this module. The immune system is composed of cells, tissues, and molecules that mediate resistance to infections. Immunity is the ability of the human body to protect itself from infectious diseases, and the collective and coordinated responses of the body against any foreign substance is termed immune response. Viruses and bacteria are everywhere. Some of them want to invade the body. But how does the body defend itself against these viruses and bacteria? The first line of defense are nonspecific and are designed to prevent infections. These include intact skin, mucus, cilia, and phagocytes. Mucus contains lysozymes or enzymes that destroy bacterial cell walls. The normal flow of mucus washes bacteria and viruses off of mucous membranes. Cilia in the respiratory tract move mucus out of the lungs to keep bacteria and viruses out. Phagocytes are several types of white blood cells, including macrophages and neutrophils, that seek and destroy invaders. Some also destroy damaged body cells. Phagocytes are attracted by an inflammatory response of the damaged cells. The immune human system is able to react to an enormous number and variety of foreign antigens and provides immunity through two complementary types of responses. Innate immunity is the body's initial defense mechanism that comes into play immediately or within hours of a pathogen's entry into the body. Innate immunity is made up of physical barriers like the skin and mucous membranes, some physiologic defenses like temperature, low pH, and chemical mediators, evolutionary conserved pattern recognition receptors that react to protein signatures on microbes, such as the pathogen-associated molecular patterns, as well as phagocytic and humoral inflammatory responses. It does not depend upon previous exposure to the pathogen, nor does it produce any immunologic memory, and it does not improve with repeated exposure to the pathogen. Why are not specific defenses enough? Why do we also need specific defenses? Adaptive immunity is the body's second level of defense, which develops as a result of infection with a pathogen or following immunization. It defends against a specific pathogen and takes several days to weeks to become protective. Adaptive immunity has the capacity for immunologic memory, thereby providing long-term immunity which may persist for a lifetime and increases in strength and precision each time it encounters a specific antigen. There are two main mechanisms of immunity within the adaptive immune system, the humoral and the cellular. Humoral immunity is also called antibody-mediated immunity. With assistance from helper T cells, B cells will differentiate into plasma B cells that can produce antibodies against a specific antigen. The humoral immune system deals with antigens from pathogens that are freely circulating or outside the infected cells. Antibodies produced by the B cells will bind to the antigens, neutralizing them or causing lysis or dissolution or destruction of cells by a lysine or through phagocytosis. Cellular immunity occurs inside infected cells and is mediated by T lymphocytes. The pathogen's antigens are expressed on the cell surface or an antigen-presenting cell. Helper T cells release cytokines that help activated T cells bind to the infected cell's MHC antigen complex and differentiate the T cell into a cytotoxic T cell. The infected cell then undergoes lysis. T cell recognizes peptide antigen on macrophage in association with major histocompatibility complex or MHC class, identifies molecules on cell surfaces, helps the body distinguish self from non-self. T cells go into effector cells stage that is able to kill infected cells. There are two types of T lymphocytes: the helper T lymphocytes or CD4 cells that activate phagocytes to kill microbes, and the cytolytic T lymphocytes or CD8 cells that destroy infected cells containing microbes or microbial proteins. Cell-mediated response can be either primary or secondary. In primary response, there is production of specific clones of effector T cells and memory clones that develops in several days, but it does not limit the infection. Secondary response, on the other hand, is more pronounced, faster, and more effective at limiting the infection. Humoral immune response involves B lymphocytes that recognize specific antigens, proliferate, and differentiate into antibody-secreting plasma cells. Antibodies then bind to specific antigens on microbes, destroying them via specific mechanisms. Some B lymphocytes evolve into the resting state, or what we call the memory cells. An antigen is a substance that the body may recognize as foreign and that may trigger immune responses. The terms immunogen and antigen are often used interchangeably. Antigens are molecules that initiate the production of an antibody and causes an immune response. They are large molecules of proteins present on the surface of pathogens such as bacteria, virus, or even fungi and other foreign particles. Upon entry into the body, it induces immune response for the production of antibodies. Antibodies are proteins that are produced in response to antigens introduced into the body. These antibodies protect the body from disease by binding to the surface of the antigen to block its biological activity, or what we call neutralization. By binding to the antigen that coats the surface of the infectious agents to make it more susceptible to clearance, called phagocytosis by phagocytes, called opsonization, or by binding to specialized cells of the immune system, allowing them to recognize and respond to the antigen. Sometimes, activation of the complement system to directly cause disintegration or lysis of the infectious agent or pathogens will enhance its phagocytosis and attract other immune cells toward the pathogen. IgM are produced as a first response to many antigens, and levels remain high transiently, while IgG are produced only after IgM, and higher levels may persist in small amounts throughout life. IgG are produced in large amounts during secondary response as a result of persistence of antigen-sensitive memory cells after the primary response. Immune response helps individuals defend against microbes and some cancers. However, there are times that the immune response can fail due to hypersensitivity reactions and immunodeficiency. Hypersensitivity reactions can cause cell damage through excessive immune response to antigens. Hypersensitivity is an overreaction to infectious agents, while an allergy can be an overreaction to environmental substances. Autoimmunity, on the other hand, is an overreaction to self. Immunodeficiency is the loss or the inadequate function of various components of the immune system and can occur in any part or state of the immune system, be it the physical barrier, the phagocytes, the B lymphocytes, the T lymphocytes, complement, or the natural killer cells. The immunocompromised host has an impaired function of immune system and is at high risk of infection. Immunodeficiency can be congenital or primary, for example, genetic abnormality or a defect in lymphocyte maturation. It can also be acquired or a secondary immunodeficiency that results from infections, nutritional deficiencies, or even diseases. And because the failure of immune response can result in hypersensitivity and or immunodeficiency, medical science has created systems to help the immune system in fighting against infections through antibiotics for treatment and, more importantly, vaccines as a preventive tool. Different types of vaccines work in different ways to offer protection. But with all types of vaccines, the body is left with a supply of antibodies that will remember how to fight that virus in the future. It typically takes a few weeks after vaccination for the body to produce these antibodies. More of how vaccines work in the next modules. In summary, the human immune system is able to react to an enormous number of variety of foreign antigens and provides immunity through either the innate immunity or the adaptive immunity. The innate immunity relies on mechanisms that are already existing even before the microbe enters the body and is often the first line of defense. However, it has no memory for subsequent exposure. Adaptive immunity develops after the microbe enters into the host and comes into action after innate immunity fails. This has memory to deal with subsequent exposure through the use of specific cells such as the T cells and the B cells. Primary immune response is often short-lasting and smaller in magnitude, while secondary immune response lasts longer and is larger in magnitude as it develops memory cells following primary response. We hope that this module was able to refresh your memory on the basic immunology